The overall goal of this procedure is to optimize polymer-coated surfaces to preserve the long-term stable culture of stem cell-derived human hepatocytes. This is accomplished by first synthesizing polyurethane 134 or PU134. The second step is to choose the right solvent to solubilize PU134. Next, the spin coating process with the PU134 on the glass cover slip is optimized. The final step is optimization of the sterilization procedure of the polymer PU134 coated glass cover slips. Ultimately, scanning electron microscopy, atomic force microscopy, and drug metabolism assays are used to show the influence of the PU134 polymer coating in the function of stem cell-derived hepatocytes.
The main advantage of this technique over existing methods, like the use of variable matrices in culture in maintaining pluripotent stem cells-derived hepatocytes, is that these synthetic materials can be fine-tuned for purpose and scaled to quality-assured standards. In addition, they display batch-to-batch consistency.
The implications of this technique extend toward therapy of liver diseases because it represents an example of how the surface of a synthetic polymer can be optimized to preserve cell phenotype. Demonstrating the procedure will be Dr. Jeffrey Walton, a postdoc from my laboratory.
Prior to starting this procedure, apply heat treatment to the monomers 1,6-hexane diol, diethylene glycol, and neopentyl glycol at 40 degrees Celsius for 48 hours in a vacuum oven to remove any residual water. Add 22 millimoles of each monomer and 55 millimoles